Good afternoon, everyone. Good afternoon, good evening, maybe good morning, depending on where you are in the world. Welcome. It's wonderful to see so many of you in the room and to see so many faces and names online as well. I want to also offer a special welcome to Dr. Nachman's family. I want to point out anybody who's here with us. Hello, 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 everybody. And also to your family and friends joining us virtually. I'm Keisha Pollock-Porter, Dean of the Bloomberg School of Public Health, and it's a pleasure to be with you for today's Dean's Lecture. This series was first established under Dean Al Somer, and it gives us an opportunity to highlight newly appointed and promoted faculty here at the school, professors and senior scientists. And we have a chance to hear firsthand how they're changing the world through the power of public health. And today, I'm so pleased to introduce our speaker, Dr. Keeve Nachman, who I've had the privilege of knowing since he was a student. So I'm excited to be here. We go back to health policy management days. And I shout out Tom Burke, who's also in the room, and all the other HPM folks. Okay, yeah, Tom gets a round of applause too, okay. All right. So Keeve is the Robert S. Lawrence Professor of Environmental Health and Engineering and the Associate Director of the Center for a Livable Future. For more than 20 years, he has been conducting groundbreaking work in the field of food systems, environmental health, and risk assessment, a journey that began at the Bloomberg School. Keeve earned both his MHS and PhD here, and it was a class taught by Tom Burke that opened his eyes to the potential of improving lives through better environmental health policies. By the early 2000s, Keeve had developed an interest in exposure to heavy metals, but was still searching for a dissertation topic. I'm sure many of you can relate. Then he attended a CLF-hosted seminar and inspiration struck. Researcher Amy Sapgoda was lecturing on industrial food production, particularly what we feed to animals. She mentioned arsenic-based drugs, which for Keeve begged a serious question. Why would we feed a known human carcinogen to chickens that we intend to eat? In that moment, he'd found not only a dissertation topic, but also a public health issue that needed attention. Over time, his research linked the use of these drugs to increase cancer risk. With powerful evidence in hand, he began advising on legislation and advocating for policy change. It took 15 years, persistence, but thanks to the efforts of Keeve and his colleagues, the US banned arsenic-based drugs in food animal production. Other countries followed suit, including China as the world's largest poultry producer. This was a major public health victory that Keeve achieved through both research and advocacy, and thanks to both his curiosity and, again, his dogged persistence. And of course, it's just one example of his decades of work to improve food systems and environmental health around the world. Keeve has also used AI to study wastewater treatment plants nationwide and identify chemicals that aren't currently on the EPA's radar. He has helped shed light on the shocking hazards of Cancer Alley in Louisiana using data from a truck outfitted as a mobile laboratory. He's co-authored groundbreaking papers recommending country-specific dietary shifts to improve the health of both people and the planet. And through his work and more, he's not only advancing knowledge, but bringing it to policymakers and the public in accessible and useful ways. In fact, Keeve's work has been featured on the Colbert Report not just once, but twice, which clearly shows he's bringing the work of public health out into the world. He's also a committed teacher and advisor who is excited to show students how multidisciplinary research and evidence-driven advocacy can lead to real change. Through all of his efforts at the school and beyond, Keeve is shining new light on the chemicals and hazards that we encounter in our everyday lives. He's addressing environmental injustice and helping us to imagine and build a healthier future for ourselves and our planet. Keeve, thank you for all you do and for sharing your work with us today. We are excited to hear from you, and when you're done, we'll open up for questions. So thank you.
Thank you so much, Dean Pollock-Porter, for that lovely introduction. You did spoiler a couple of my case studies, but maybe the detail I provide today will liven it up. Yeah, no, it's really amazing to be here, and it just warms my heart to see this room. thank you for coming. It's so cool. So anyway, thank you also to those folks online for joining us today. Looking forward to sharing my career with you. So in preparing for this, I watched a couple of other dean's lectures, and they blew me away. Like, what an amazing faculty we have here at the school. But I found myself with the challenge of, like, what am I going to talk about? I worked on crazy random set of things, or said in a different way, I've had the opportunity to address a lot of urgent public health crises. And so I needed to figure out, how am I going to tie my body of work together? And what I came up with was, I want to talk about the space between the research and the action, because I feel like that's the part of this whole process that I'm most passionate about. And I want to find a way, anytime we write a paper or come up with some scientific discovery, that it finds its way to actually making change to make the world a better place, to make people healthier, and to make livelihoods better. So that's the theme for today. And I'm going to dive into some of my work. It's going to be a wild ride quickly through a lot of things. And I want to acknowledge that there are things that I've worked on with people in this room that I'm not able to get to today. And please don't feel slighted. Every project has a special space in my heart. Okay, so let's get going. Oops, there we go. Here's a figure depicting my career trajectory. Probably caught that from before, but I want to just kind of tell you how I got to where I am today. And I'm going to go way back. So this is 16-year-old Keeve at Fork Union Military Academy in rural Virginia. I started here and I applied to Johns Hopkins right out of military school. And through some miracle, I got in. I'm still blown away by the fact that they admitted me. Maybe I was different. Anyway, when I got to Hopkins, I talked to my new advisor and I said, hey, you know what? I would love to major in fiction writing, and I also want to be a physician. And he said, that's an interesting combination, Keeve. Why don't you start with fiction writing, and we can get to all that medical stuff later. So I learned pretty quickly that I wasn't a very good fiction writer, and I needed an alternate path. And so I still had that interest kind of in medicine. And so I got a cool job in the Department of Medical Physics, working with a guy named John DeCello. And John wrote me in. I was looking for a job, $8 an hour. But he had this really cool project that was aimed at trying to understand how certain therapeutic drugs like tamoxifen and raloxifene could be used to prevent cancers in astronauts who were exposed to cosmic radiation, because it takes about 1.5 years to fly to Mars, and this was to prepare us for an eventual trip to our neighbor. And being involved in that study was fascinating, even though the labor of it was a little dry. And it made me think, you know what, maybe I'm interested in not only medical physics, but research. And so I applied to the Department of Environmental Health Sciences. We now have a new name. I couldn't find the EHS logo. And I worked in I did my degree in radiation health sciences, which no longer exists. I may have been the last radiation health sciences student. And I started the master's program here, the MHS. And in my first week, they made me take this class from this guy named Tom Burke called The Introduction to the Risk Sciences and Public Policy. And I had no idea what it was, nor did I have any interest in that class. And it was right after my 3-hour radiation class. And I went a couple times, and I started to think, this public health stuff, this environmental health policy stuff, I kind of like this better than medical physics. And the idea of doing work that impacts people millions at a time is much more attractive than zapping a tumor at a time. Not that that's not very important. So talked with Tom. He hired me for the summer. And after working for Tom for a year, I thought, you know what? I think this is for me. So I talked with Tom and said, hey, I want to be your PhD student. And I applied to the Department of Health Policy and Management. Very important, informative experience for me. And was a classmate of Dr. Pollock Porter. And in the course of doing my PhD in HPM, I had the opportunity to apply for a Center for a Livable Future Fellowship, and I was accepted, which was great. It's where I first met Sean McKenzie, and I also met a man named Bob Lawrence. And I'm going to come back and talk about Tom and Bob in just a moment, but meeting the two of them was really important in directing my career trajectory. So after I finished my PhD, I put out a couple of applications and ended up getting a job at the National Center for Environmental Economics at the EPA, which was a really, really important and formative experience for me, because I learned how science was used to enact regulation to protect people from chemicals and other environmental hazards. After that, I had a brief stint with the Department of Defense and the Army Corps of Engineers and learned that government was not for me. So after that, I reached out to my colleagues at the Center for a Livable Future and said, hey, got anything for me? And they ended up hiring me back as staff. I was the science director. And then in 2010, I became an assistant scientist in environmental health and engineering and applied for a tenure track position in 2014, became an assistant professor, and here I am today. Also throughout, I've been involved with the Risk Sciences and Public Policy Institute. I was even the co-director of the Institute for a period of time. And those methodologies, as I'll talk about, are a big part of my work portfolio. So I want to pause for a minute and recognize Tom Burke, and I hope Bob is online. If not, Bob, when you see this, but Tom and Bob continue to be a guiding light for me in my career. What is really notable to me about both of these guys is that while they're terrifically accomplished scientists, where they really shine, where they really stand out, and what I believe they're going to be known for, what they're known to me for, is their practice and advocacy portfolios. They have made public health work in the real world. And between being the assistant administrator of the Office of Research and Development at EPA and starting the Center for a Livable Future, both Tom and Bob have had indelible impacts on public health. And both of you inspire me. So anyway, I'm going to dive into my work now. When I got started in public health, a lot of my work was on the issue of industrial food animal production. And I'm going to show you a couple pictures just to kind of quickly orient you to the world in which I'm operating. So this is an industrial feedlot. This is an industrial turkey production house. This is a small swine CAFO. And this is an industrial poultry operation. And what all of these things have in common is animals being raised in tight proximity, in very unhygienic conditions, and posing risks to the people who work in them, to the people who live near them, and to the people who consume food produced in them. These are the basis for the start of my career. And so the first issue I worked on, and the dean mentioned this, was a chemical called roxarsone. Roxarsone is an arsenical drug. That's a picture of the molecule for all you chemists. And roxarsone is an anti-parasitic drug that also makes chickens, turkeys, and pigs grow faster. And it's been fed to animals since the 1940s. And as the dean said, when I first heard about this as someone who was studying heavy metals and risk assessment, I thought, that's freaking crazy. Why would we feed a known human carcinogen to an animal that we plan to eat? And so anyway, just one quick allusion back to military school is that I knew even back then that I wanted to work on arsenic, and I was even getting my name ready for how it would appear in PubMed. Okay, that's the last military school photo. I'm very sorry. So anyway, when these drugs are used, there are two ways that people can be exposed to arsenic. And that's not a good thing. Does everybody know arsenic is bad? Do I need to litigate that again? So arsenic is a known human carcinogen and linked to all sorts of non-cancer health effects. So you don't want to be exposed to arsenic. So when we use these drugs, arsenic, most of it actually gets into the chicken waste, and then we have to manage the chicken waste, right? And so we usually apply it to farmland as fertilizer, and that poses all sorts of problems in its own right. And the other way that people can be exposed to arsenic is when they eat meat from animals that are treated with this drug, right? And so thinking logically about the majority of the problem being in the waste and in the environment, that's where I started with my work on this drug. And I published a series of papers on it. I want to show some of the people and some of the topics I covered. The first thing I did, first paper ever, was a commentary, and this turned out to be a really, really important paper in my portfolio. It's weird to think a commentary matters so much, but it's a straightforward communication of what we know. But anyway, I worked with Ellen Silbergeld, Lance Price, and Jay Graham to discuss the public health implications of burning poultry litter that was loaded with arsenic because of the use of roxarsone, and how when you burn arsenic, it doesn't go away. It either goes out the stack of the incinerator or it ends up in the ash that, guess what we do with that? We spread it on agricultural land. So we're recycling arsenic back through the food system or introducing it into the air. That's a bad idea. That was my first paper. Second one was my dissertation work, which I worked on with the late Allison Guy, Jana Mahalik, who I think is online. Hi, Jana, and Tom Burke, where we looked at an intervention to reduce the loading of manure on farmland and getting into aquatic ecosystems and causing eutrophication and all sorts of other things, to pelletize it and then sell it as a home and lawn garden fertilizer. The problem, of course, being arsenic, and that by dewatering it and turning it into pellets, we're then depositing arsenic-loaded pellets into residential and commercial settings. They were used on golf courses, so that was also a bad idea, and we documented the extent to which people would be exposed through the use of those pellets. And then lastly, and this is the weirdest one, this is poultry feather meal. So most of us don't eat chicken feathers, I guess. And so when chickens are being processed, we remove all the feathers, we heat them up to kill bacteria, and then we chop them into this fine powder. Can anyone guess what we do with it? We feed it to animals and we sell it as organic fertilizer amendments, okay? And so in feeding chickens arsenic, there was a concern that we had that feathers are a lot like our fingernails, and fingernails and toenails are great biomarkers for arsenic exposure. So we assumed that the arsenic would travel into the feathers, and sure enough, it did. So those papers were important in spurring action, actually. So after I left the DoD and came back to the CLF, within 1 week of being here, this was surprising. I hadn't thought much about roxarsone. I'd kind of moved on to my next series of topics. And I got a phone call from a guy named Mike Ryan, who was the legislative director for Congressman Steve Israel. And I was shocked. And he said, hey, Keeve, we've seen some of your papers, and the congressman wants to introduce legislation to ban arsenic-based drugs. And my head was exploding. I thought, A, someone read my paper, really? And then B, they want to actually do something about it? That's amazing. And he said, so Keeve, would you be willing to advise the congressman on this issue? And I was like, sure, having no clue whatsoever what advising a congressman means. And so let me tell you what it means. So advising a congressman in large part meant enduring meetings with industry stakeholders he'd ticked off by introducing the legislation. So I, on a regular basis, would take the train down to DC and go to his office in the Rayburn building and be in these really hostile and intense meetings with people who wanted him to drop the legislation altogether. It was kind of soul-killing. The worst of those meetings, I remember I got a call the night before from the legislative director, and he said, Keeve, need you to come in tomorrow. We have a meeting. Just wanted to warn you, this one's going to be unpleasant. And I said, Mike, they've all been unpleasant. What's different about this one? And he said, well, we've got one of the more notorious lobbyists for the meat industry named Steve Copperwood. He's defended cockfighting and all sorts of other horrible things, and he is not a nice man. So just mental preparation, it'll be fine. And I said, great, thanks, see you tomorrow morning. And I tried to go to sleep, didn't sleep well. I remember the train ride down, just worrying the whole time. And I remember the rainy walk from Union Station to the Rayburn building. And I got in there, and he brought his whole entourage of lobbyists with him. And he had this big stack of papers. And he came in and he slammed them on the desk. Whoa, that was harder than I meant. It was my dissertation. And I was so ready for him to just intimidate me and browbeat me. And it turned out he had nothing, and I totally kicked his butt. But the bravado was important. And so anyway, that was a series of meetings for sporadic periods over 2 years. I also had the opportunity to do my first press briefing. And you can see me there. I didn't have any gray hair yet. And you could see the congressman carrying around a cooked turkey. This was around Thanksgiving time. It felt like a charade a little bit. I read my very nervous statement. I was very, very, standing up straight, had advocates around me, appreciated them being there. And I was going to talk to reporters after, and I'd never really done that before. And I remember I'd memorized the arsenic mechanisms of toxicity, and I knew all these minutiae. And they came, and I was like, I'm ready for your questions. And they're like, what's arsenic? And I was like, So I had to quickly learn the way I think about things is not really necessarily the way other people think about things. And I got to better prepare myself for the audience. So that was a great learning experience. Sadly, after 2 years, 2 sessions of Congress, the congressman called me and said, Keeve, I really appreciate all you've done working with us on this, but I'm getting so much opposition to this bill, and I'm not getting anyone to take it up with me, and so we're going to have to let it go. Doesn't mean we won't bring it back in the future, but for right now, it's dead. And so the bill died. So that was a bummer. But thankfully, following that, two legislators in the state of Maryland, Tom Hucker and Paul Pinsky, introduced similar legislation in the state of Maryland, which was a big thing because Maryland is a big, big poultry state. So for them to do this, it was a risk. And they also asked me to advise their office, which I happily did. I felt a little more emboldened and knowledgeable, ready to tackle it on a smaller stage. I did get my first opportunity to give testimony in front of a legislative body. This is a picture from one of the times that I did it. And I remember the prep for this. They're like, Keeve, you can't read it. You got 3 minutes. You got to cover these things, and you got to be good. And so anyway, the day is there. I have my testimony. I've written it out. I've memorized it. I'm so ready to go. And they held our bill until the end of the day, which apparently is what they do when they have something contentious, because they want people to get sick of staying there and leave. And I think our bill started somewhere between like 8 and 9 o'clock. And I remember I got up and I'm so freaking nervous and I'm ready to do this. And the chair of the committee's like, I'm so sorry, we're really tired. Do you think you could just do a minute? Like, we get it, okay? And so I had that enjoyable experience. But that wasn't the most memorable part of this, unfortunately, because as soon as I did my testimony and the panel of proponents for the bill to ban arsenic had gone, then there were 5 panels from the opposition, and they were from the industry. And they were all people with PhDs and veterinary degrees. And they were all about my dad's age. Sorry, Dad. And they got up one by one and essentially pointed at me and said, he's wrong. And I wasn't wrong. They were wrong. But the industry had brought its full force in opposition of this bill because of potentially what it meant for them. But I got to tell you, getting up as a junior scientist and watching very senior people really try and push back against you, it was kind of soul killing. I went home that night and I was like, screw this. I'm going to just write journal articles. And I thankfully, Becky was there and helped coddle me through. And I woke up the next morning and went to the other chamber of the Maryland legislature, and exactly the same thing happened, except for this time I was a little more ready. And then at the end, I had Pinsky and Hucker pull me aside and say, Keeve, yeah, that was painful, but I got to tell you, this is how it works. We rarely win the first year, and we got to do it again. And so we did it a second year, and we did it a third year. And the third year, I had some colleagues from Food and Water Watch who had put together this amazing grassroots campaign to change public opinion about this and put pressure on the legislature. And each year, the industry opposition weakened, and the public health pressure to do this increased. And finally, we won. And so we were able to ban I keep clicking this. We were able to ban arsenical drugs in Maryland for the first time. We were the first state in the union to do it. And so it wasn't everywhere in the US, but it was a really important start. And it's not always the case that we get a win in public health. So that felt really good. Okay, so while this stuff was happening, I was still doing research too. And so I'd found that maybe the waste is not the angle to getting this changed, because when arsenic and waste pollutes soils and potentially pollutes groundwater, it affects people who live in rural areas, but it doesn't affect a lot of people, right? They kind of can ignore what's happening to people outside of their own domain. So a lot of people eat chicken, so let's look at what happens to chicken, and maybe that's a basis for intervention or for change. So with some generous support from the Center for a Livable Future and some terrific colleagues at the CLF, we did a market basket study of chicken products to test them for this drug and inorganic arsenic. So we went to grocery stores in 10 cities around the country. I got to take a lot of great trips and spend a lot of time in grocery stores. And we bought organic, we bought conventional, and we bought antibiotic-free chicken. We took each breast of chicken and cut it in half and cooked half, left the other half raw, and then tested them using ICP-MS to measure different arsenic species, which is just a fancy word for saying different chemical forms. So we could figure out when using these drugs happens, what happens to the arsenic? Does it stay behind in the meat? And does it expose people to inorganic arsenic, a known human carcinogen? And so here's some of the results. I'm going to go a little fast, but one of the challenges in studying animal drugs is that package labels generally don't have to tell you what drugs were used. Organic means something, right? It means most of these pharmaceuticals can't be used, but if there's no label on it, it really doesn't indicate anything about whether drugs were used. So we sliced our data 3 different ways. The first is, what was the package label? Was it conventional? Was it antibiotic-free? And was it organic? And so conventional is the most likely status for using the drug, and we saw conventional was a lot higher. Then if we broke the conventional out into packages of chicken where they said we don't use arsenic, because that was a thing then. There were actually brands where they would say we do not use arsenical drugs because it had garnered some public attention, we saw an even bigger divide. And then lastly, in a sample of chicken where we measured the parent compound, the original drug that was fed, we found the highest difference in signal in inorganic arsenic. And so taken together, this means that when the drug is used, it likely increases people's exposure to a known human carcinogen, which is not a good thing. And this FDA safe limit here is a level that they'd published in one of their documents to say if it's above this, it's violating the Delaney Clause. So anyway, what does it mean when people actually consume chicken? That's my favorite picture in this whole thing. So jumping ahead, if we did some risk modeling based on what we know about arsenic and what we know about poultry consumption, we estimated that when this drug was used, the increased population lung and bladder cancer burden over a 70-year period is 8,661 cases. That's a little harder to interpret for most people. So on an annual basis, it means using this drug could lead to 124 cases of lung and bladder cancer per year, solely as a result of the decision to use this drug. So in my opinion, that's a pretty compelling case for why this drug, which we could elect not to use, should be banned. And so I was really fortunate to have our work covered in the New York Times by Sabrina Tavernisi. Anybody know her? She did the Daily podcast. Lovely person. I've had a chance to work with her a lot. And she did an exclusive. So excited for this to come out and get covered. And I also had the opportunity to work with a number of NGOs who were interested in a lot of the type of issues that I worked on, and they had actually sued the FDA over a latent petition to ban arsenical drugs. And when my study came out, they amended their lawsuit. The lawsuit had nothing about dietary exposure risk, but now that they had these data, they were able to integrate it into the lawsuit, and it mattered. So FDA announced on September 30th, 2013, that they had received word from the sponsors, the companies that manufacture 3 of the 4 arsenical drugs, that they were going to withdraw the approvals, effectively taking them off the market, which was a big deal, big win, meaning they were illegal to use in the United States. They did leave one drug that was very similar, allowed to be used, which was kind of a bummer at the time. But thankfully, we were doing additional research on that final drug. It's called nitrazone. And we did another market basket study and found very similar things, and did another biomarker study using NHANES data that showed that when people ate poultry products, their exposure to arsenic was indicative of their urine arsenic levels. And taken together, this formed the basis for the FDA to then act 2 years later and ban the final arsenical drug, meaning that in the United States, as of 2015, these drugs were illegal. And thankfully, other countries followed suit. One of the most exciting countries to follow suit was China. China produces 5.3 billion chickens a year. They are the world's largest poultry producer. And so the benefits that we estimated in the US are far smaller than what we actually see in China. China also exports a lot of poultry. So this felt like a big win. We don't always win in public health. And this was a good one. So these drugs are on the way out entirely globally. So I whet my appetite on arsenical drugs, but there are a lot of other issues related to drugs and animal agriculture that I thought were worth tackling. So the next one learned a lot from Bob Lawrence, Lance Price, Ellen Silbergeld, and many others, was this issue of antimicrobial resistance resulting from feeding food animals antibiotics on a regular basis. So why does that matter? So I am having some difficulties. I'm going to test my professor slideshow prowess. Let's see. No? Spacebar? Look, I got it back. All right. Yes. Okay. So we feed animals antibiotics on a regular basis, often at very low levels, often for their entire lifespans or close to it. And the problem with doing that is that antibiotics are really important in human medicine. And unfortunately for us, the industry uses a lot of the same drugs or classes of drugs that we would also use to treat infections in people who are sick. And so what happens when you feed these drugs to animals or you expose bacteria to them is that all the susceptible ones, which are the purple bacteria here, they die. But the teal one is resistant in this example, and it survives. And what happens is that that teal bacterium is able to take advantage of the microbial ecosystem and reproduce. And so there are no more susceptible bacteria here, only the resistant ones. And resistant bacteria are bad. right? And so they sadly don't stay on the farm. And when animals leave the farm, or when we manage waste from animals on the farm, we're able to spread these resistant bacteria into the environment and potentially to humans. And when a person gets infected with these bacteria, those infections are incredibly difficult to treat. They're more expensive to treat. They take a long time. And sometimes people die from them. Often people die from them. So it's a really serious public health problem. And it calls into question, why the heck do we routinely waste these drugs on food animals? So I wanted to work on that. That was a pretty cool thing. So remember the feather meal I showed you? So part of that experiment wasn't looking just at arsenic, but it was looking at, could we find residues of some of the other things we feed food animals in the feather meal? And remember, I mentioned before that we use feather meal as an animal feed amendment and also as an organic fertilizer. And so when we did our panel to look at what was in the feather meal, first thing is we found arsenic, I mentioned that before, but we also tested for residues of antibiotics. And what we found was kind of shocking. So we found across our samples between 2 and 10 different classes of antibiotics in every single sample. Which was really shocking. And the most shocking thing that we found was we found residues of a drug called enrofloxacin, which is a fluoroquinolone drug. Those drugs had been banned from use in poultry for more than 5 years at the time that we acquired the samples. So it was potentially indicative of using drugs that were no longer legally allowed to be used in this type of food animal production. So that was quite upsetting. We also, because the panel we were using also had the ability to detect some human pharmaceuticals, decided to take those data as well. And we found residues of diphenhydramine, acetaminophen, and fluoxetine. I can't believe I said those 3 drugs in rapid succession like that. But it was suggestive that the animals were fed those compounds as well. Those were found in 2 samples that we acquired from China that were commonly sold for use in the US. So anyway, this suggests, especially on the antibiotics front, that some of these antibiotics, through the use of feather meal, are being cycled back into the food system, both through direct addition to animal feed, and also if you're using organic fertilizer at home. And so I had a chance to talk about this work with Nick Kristof from the New York Times, which was really cool. He wrote a piece about it, and it ended up being the #1 forwarded opinion piece or even news article in the New York Times that day, which was really cool. And I ended up doing a media frenzy. This is my first time doing a TV media frenzy. I got this amazing picture of me. This is the worst picture of me ever taken. But I still didn't have gray hair. But it was a great opportunity to talk about this and raise the public consciousness about the issue of using or misusing antimicrobial drugs in animal production. And so, a lot of good coverage, and the media fervor died down. And I was moving on to the next project. And about 2 weeks later, I got a phone call from a colleague, and I just dropped off my kids. And she said, "Hey, Keeve, you finally made it." And I said, "What are you talking about?" And she said, "I guess you didn't watch Colbert last night." But Stephen Colbert became aware of our study through the Nick Kristof column and ended up doing a 4-minute bit about our study, which is the wildest thing I've ever seen, and a career achievement I will never top. So that's the end. You guys can leave. I'll see you later. Actually, let's keep going. So media attention is important. It's definitely part of raising awareness of an issue, but advocacy takes a lot more than that. So one of the cool things I've really enjoyed about being at CLF is taking science and taking public attention and harnessing it to influence the activities of federal agencies. So we wrote so many letters, and I did this with my colleagues Tyler Smith, Bob Martin, and Bob Lawrence, trying to pressure the FDA to get more aggressive with its restrictions on antimicrobial drugs. We also published a couple of keenly placed letters in the journals Science and Nature, calling for more stringent FDA action, which we thought was helpful. And what the FDA ended up doing, it was imperfect, but it likely had some impact, was they introduced something called Guidance 213, which restricted certain types of uses of antibiotics in animals. In particular, they banned the use of antibiotics for growth promotion, so to make chickens grow faster and fatter. Same thing with pigs. What they allowed, though, which was a disappointment, was that these drugs continue to be used for disease prevention. Disease prevention sounds pretty awesome, right? Who doesn't want to prevent disease? However, on the ground, in practice, what that means is the use of these drugs routinely at low levels without any sort of idea of what bacterial target they were trying to hit. So really, it was very similar in effect to growth promotion. So that was a bummer, but Thankfully, some of the states in the union tried to pick up the baton where the FDA left it off. And the first state to pass a law to do better than what the FDA did was California. Not surprising, right? California is often much more progressive with passing public health regulation. So they passed a bill called SB 27. And the idea behind SB 27 was that it was supposed to address this disease prevention loophole and disallow that. They were also supposed to track the use of antimicrobials in food animal production for the first time at the use level. So farms were supposed to report use. So we, and by we, I mean a terrific team of people, some of whom are here today, put in a couple of grants, one to the Fisher Center here at Hopkins to get some pilot money, but then to the NIH and the Wellcome Trust to study this natural experiment of the passage of this bill in California. And we published a series of papers. The first one was looking at the data collection that was occurring under the SB 27 bill, and much to our dismay, we found out that the California agency responsible for collecting information about antibiotic use reported data in a way that made it impossible to infer any changes in antibiotic use as a result of the bill. So that was disappointing. But we were actually able to use data from field collected samples of animal products and also isolates of urinary tract infections with E. coli to assess whether the bill was having any impact on rates of resistance among infections of people living in California eating the chicken there. And we did something really cool. This was led by Joan Casey and Kara Rudolph, who were part of our team. We did a synthetic control analysis where we created a synthetic California. So California, in reality, passed a law that's supposed to change the way these drugs are used. We created a synthetic California that did not pass a law. And we compiled that from information from other states. And we were able to show that for one class of drugs, the extended spectrum cephalosporins, that we actually saw a reduction in resistance following the passage of the law, which is not definitive proof that it worked, but it's suggested that the passage of that law may be having some effect in reducing drug use and reducing resistance. So that has huge implications. And I wanted to show another paper that was led by Lance Price, who's here today, looking at the E. coli that we acquired during that study. And he was able to use genomics to determine that among our sample in California of people with urinary tract infections, about 20% of them could be tied back to zoonotic sources or animal sources. And that rate was even higher among people of lower income. So that illustrates that this phenomenon of resistance is playing an active role in people's infections, and it's not just hospital sources or other vectors. So that was really important in moving the antibiotic issue forward. And we are by no means done with that issue. There's still a lot to do, but I wanted to transition to another topic. So I love drugs. Sounds weird to say out loud. But I also wanted to work on other issues in the food system, and I've had amazing opportunities to do that. So I wanted to talk about climate and the complex interrelation between climate change and agriculture. So it's pretty well recognized that raising animals for food, producing food, has a climate footprint. And so I had the good fortune of working with this amazing team of people, again, some of whom are in the room, to take a really nuanced look at what opportunities we may have to change diets and how those changes in diets may affect the release of greenhouse gases and may mitigate some of society's impact on climate change. And so what we were able to do is take data about dietary patterns, about trade, and about the greenhouse gas and water intensity of various food products, and model how dietary shifts in 140 countries around the world create opportunities for mitigating some of our global environmental catastrophes. And so this is a very busy figure. I'm going to tell you about each dot on it, just kidding. But the idea here is that looking across 9 increasingly plant-forward diets, we can see how on a per capita basis on the left, by country on the bottom, adoption of a particular dietary pattern may reduce a person's greenhouse gas emissions attributable to what they eat in a year. And then on the right, it's very hard to see, but you can look at, based on population sizes and diets in those countries, how there are certainly opportunities in some countries where you don't have to go vegan, right? So maybe something short of that decreases the amount of animal proteins that are tied to really heavy-duty greenhouse gas intensity can yield dividends for mitigation of greenhouse gases releases and positively affect the climate, maybe avert catastrophic climate change or be an important part of that. So that was a really cool project to be a part of. Then I wanted to transition to something we're working on now. Just published our first paper from it. This is related to the issue of biogas or manure digesters. So the idea behind this is when we raise cows and pigs, they generate a lot of waste. Hopefully that's not a surprise to anyone. And in those two species, when we handle their waste, we do it in liquid form. We store it in these enormous cesspits. And the industry, as a way to generate energy, has started to build these things called manure digesters, where they put a cap over the manure lagoon and they try to harvest the methane from it. And on its face, it sounds like it might be a good idea, but there are many reasons to believe that there could be new issues associated with linking a new energy system to an existing industrialized food animal production system that already has massive public health decrements associated with it. And so we published a paper where we took all of the arguments for and against manure biodigesters that are made by companies that want to build these things and also advocates that are fighting against them to try and take the existing evidence and validate or refute those claims. And so we just published that commentary, and our bottom line conclusion from it was that we found that based on existing evidence and what we know about them, these things don't seem like the climate solution that they're purported to be. And in a similar and related project, and this is something I'm really, really excited about, we worked with colleagues like Dan Ho and Frank Curriero to use artificial intelligence and deep learning to identify the locations of concentrated animal feeding operations all across the state of California, as well as these manure biodigesters. And we wanted to see if we can answer questions about whether adding biodigesters is associated with the further intensification of the food animal production infrastructure in the state of California. And we found that 3 years post the decision to add a manure digester, we found about an 800 plus cow increase per industrial dairy that's adding one of these farms. So this is suggestive evidence. This is a preprint, only preprint I'll show today. This paper's under review, but this is suggestive evidence that yes, this new type of addition to the food animal production industry is resulting in added intensification. So that was a cool finding. So not only does agriculture affect climate, climate can actually affect agriculture as well. And so anybody who's a big fan of arsenic research is well aware that rice is kind of the poster child for foods that take up arsenic. And so I was lucky to work with a man named Lou Ziska and another guy, Lou is from Colombia, Chunwu Zhu is from the Chinese Academy of Sciences. And Lou and Chunwu have this amazing system where they can simulate the effects of climate change in a flooded rice production setting. And so what they did is they grew all these different cultivars of rice and they simulated what things are going to look like in 2050 if we don't mitigate climate change. And so they're able to increase the temperature and they're able to increase the concentrations of carbon dioxide in the environment. And what we found is that by doing so, we saw a synergistic impact between temperature and carbon dioxide concentration on the accumulation of arsenic as a result of changes to the soil biogeochemistry, which is important because when you actually take that out and model people's dietary exposure to rice and inorganic arsenic, we find that those changes lead to additional millions of cases of lung and bladder cancer among rice consumers across the globe. And rice is a really, really important food. By no means are we suggesting you shouldn't eat rice. Rice is a dietary staple for nearly half of the world. So any steps we can take to mitigate climate change or to make alterations to rice production practices to limit the accumulation of arsenic are desperately warranted so we can avert these increases in cancers over the next 50 years. Okay, transitioning from food for a little bit. The tool I use is risk assessment. That's been a big part of what I do. It's an amazing decision-making tool that informs risk management and environmental policy. And we have moved from a very reductionist approach to a broader recognition that people are contending with many chemicals, many non-chemical stressors all at the same time that can compromise their health. And we need to be accounting for this diverse cascade of stressors on people if we really want to protect the public. And so the first project in this is focused on a vulnerable population. I feel funny calling my kids a vulnerable population, but I'm talking about children. So this is a shot from when my kids were little, and Becky and I was junior faculty, Becky was a graduate student, and the kids would wake up at 4 a.m. and we were tired, and we would just kind of be in their presence. But the first thing they would do every morning is they would go into the cabinet there on the left, and they would take cereal out and dump it on the floor, and then eat it off of the floor. And it's okay if you want to judge our parenting prowess. They turned out okay, but it begged the question, what are kids exposed to when they're eating dust, and how much dust are they getting in their mouths? And this has been a longstanding challenge in the field of risk assessment and exposure science. It's one of the most difficult environmental media to estimate exposure to. And if we can't estimate exposure to soil and dust, it's difficult to enact certain types of regulations like lead standards that are adequately protective of children. So I was elated when the EPA said, hey, we're going to put out an RFP. We want to fund research to better understand children's soil and dust exposure. And Sarah Leupold and I, who's here in the crowd, jumped on it and wrote a grant and got funded by the EPA and put together this amazing multidisciplinary team. I want to tell you really briefly what we did. So we essentially did 3 studies in one. We tried to figure out, where do children spend time and what are they doing in those various locations. We tried to understand their macro activity patterns as a basis for exposure modeling. And we developed a really nice data set that we just published. The second piece of it was a computer vision oriented study. So we went into children's homes, set up 4 cameras around them, and filmed them playing. And our purpose of doing this was trying to understand their rates of micro activities, which are how often they stick their hands in their mouths or how often they stick objects in their mouths. And that's really important in tandem with the larger activity patterns, so we can prospectively model soil and dust exposure. And yes, it was uncomfortable asking people if we could come into their home and film their children, but we eventually were able to convince people that it was worth doing. This is a shot from the model operating in action. There's a whole lot more behind the scenes, but if I was savvy enough to have a video, you would see actually when the child put an object in its mouth, the computer model would tell us this. So we were able to estimate how often that happened and the duration of mouthing behaviors. And prior to our computer vision study of this, there were a small number of studies that did it, but they usually involve very few children because annotating these videos takes a lot of time. So having a model that could do this was really, really cool. And then the last piece of this also involved going into homes, and I have some of my wonderful team here, and we did an environmental and biomonitoring study where we vacuumed dust from homes, collected some other environmental media. We collected dirty diapers, which was another odd ask that people were a little suspect of, but and then we collected food and breast milk. And the purpose of that was to try and identify a chemical that we could measure in either urine or stool that would be a great biomarker for exposure to soil and dust. And this was done in Carson Press's lab. It's actually still going on. So we are really excited to propose what one of these decent tracers would be. And if we can do that, that would be kind of another more top-down way of figuring out rates of exposure to soil and dust, which are going to make our environmental regs a lot stronger and more protective. I'm going to transition to my last case example now. This is Cancer Alley. A lot of people have heard about it, maybe some haven't. This is one of the most heavily industrialized petrochemical complexes in the United States, if not the world. What's troubling about this, among many things, is that lots of people live nearby. And you can imagine that the people who live nearby don't have a lot of choice in the matter, don't have a lot of money, don't have a lot of resources to leave, and have very little control over what's in the air that they breathe. And so one of my motivations for getting into this field is protecting the most vulnerable and the people who are dumped on, who are the most disadvantaged. And it just makes my heart ache to see that people live in a community where this is a common sight when you move around your community, facilities emitting all sorts of chemicals. And so I was really elated when I met Pete DiCarlo, who I think is here. I thought I saw him. Pete, are you here? There's Pete, who has this fancy toy. That's insulting to call it a toy. He has a chemistry lab on wheels. It's the most amazing thing. And he can drive around and in real time measure hundreds of chemicals over space. And what we did is we took Pete's amazing van and drove around a couple of petrochemical complexes in different parts of the country and developed these fancy geospatial maps of different hazardous air pollutants that are carcinogenic or known to elicit other diseases. And my part in this project with Pete was I would take the data from the mobile lab and try and translate it into what it means for people's health. And so with the help of my terrific doctoral student, Andrea Chigger, we developed something called the multi-effect toxicity database. And I don't have time to go into it in great detail, but the idea behind this is that we account for all of the different ways a single chemical may put stress on different parts of the body. And then we can collectively account for the mixture of chemicals working together to elicit disease across all the different organ systems. And when you overlay that toxicity database with the exposure data that Pete generates, we have the ability to estimate how likely people are to be at increased risk of different diseases. And I want to show you how this method we developed works as compared to the traditional way regulatory agencies do risk assessments. So this column here on the left, sorry, I'm showing actual data, they're hazard indices, and they're easy to interpret. If it's above 1, it's bad. If it's below 1, it's probably fine. So if you apply this approach using the traditional risk assessment way in our first case study, you find that none of these organ systems have hazard indices above 1, which would be interpreted as, no problem, let's move on, no need to do anything. But if you use our multi-effect toxicity database approach and you account for all the different parts of the body each chemical may affect, we actually find that 5 of the organ systems have hazard indices above 1. And the risk management conclusion that would come out of such an assessment would be very different. This would be a location or a population in need of some sort of intervention to minimize their exposure. And so we were able to talk about this pretty broadly, and we're covered in a couple of high-profile publications, which was cool. And again, I love media attention, don't get me wrong, you probably picked that up. But what's even more cool is that other scientists have started to take notice with this methodology. And actually just yesterday, I was invited to brief the Bay Area Air District Advisory Council and the California EPA on our methodology. And there was actually really strong display of receptivity to try and find a way to use our MetDB approach in new air pollution regulations in the state of California, which would be big. California is the test lab for the rest of the country. And it's just so exciting to see that what we're doing could be used. And I really sincerely believe that this kind of advancement in air pollution regulation could save many lives. So anyway, that's all the science. Just a couple of quick reflections. Some of the things that I've learned over the course of being here, and it's been a long time now. I hope I've made the case that discovery plus action is how we get public health wins. I love publishing, it's fun, I'm a writer, I like writing stuff, but I also know talking about it with people and finding the people who can make change with our evidence is a big part of what we have to be doing. I've also learned that doing this takes a really long time, and it often means doing stuff that scares the heck out of you. And I've learned that the scariest things are the most valuable things. It's just the way it is. As a former fiction writer, I'm not writing fiction anymore, but I do see the value in telling a story and making sure that people can understand it and they remember it and they're moved by it. And the people are the most important part. I just seen so many amazing people in this room, and I've had so many outstanding collaborators. There's nothing better. And this is such an amazing place to do this type of work. So looking forward, I have a vision for the food system and beyond and what I'll be doing in the future. But a lot of it's actually informed by a guy named Sid Lerner, who is one of the major benefactors of the Center for a Livable Future. And he was the founder of Meatless Monday. He came up with Meatless Monday. He was an original madman. So remember that TV show? He was one of those guys. And he was a marketing genius. And he would always say during my early conversations with him that marketing is something that we should be doing. And I was like, yeah, Sid, okay. I didn't get it. But I thought a whole lot more about it. And now I understand that marketing, especially in a time like now, when public health is under attack and we're not seen as people who have a lot to offer in advancing societal well-being and prosperity, we need to market ourselves, and we need to market the value of our science in keeping people healthy and making their lives actually better than they are now. So I think that's something we need to do. We need to stop being reactive and be more proactive. I've built a career out of finding problems and yelling about them, but I think we as public health scientists, as environmental health scientists, can find ways to help design and hone in on what those solutions are that will actually improve the food system and improve public health more broadly. I think we need to create opportunities for inclusion. I've found that broadening the table and inviting more people who have different perspectives and different backgrounds makes for a richer conversation and an easier way to identify better solutions. It's so important. I think it's a dark time right now, but I do think there are good things coming, and I think we just need to wait it out and stay true to our values, and I think things are going to swing the other way. I really believe that. And then lastly, I want to set the stage for our trainees, the people who are coming up next, the people who will be doing Dean's Lectures in 10 years. They need to understand the importance of advocacy and practice in realizing our public health vision. It's absolutely essential. So with that, I want to honor my students. All this work wouldn't have been possible without them and my collaborators. And I'm really excited about our Christmas party on Friday, guys. But they mean a lot, and I think we have a bright future ahead if these are the people that are going to be leading the charge. I've had the good fortune to collaborate with a zillion people, and I'm so panicked that I'm not including people. So I did some and mores, even though they're getting cut off at the bottom. But this isn't about just the scientists. I want to recognize all the people as part of our family here at Johns Hopkins that help make this work. Our fine admin team, our communications team, our education team, we all work together in really realizing our public health promise. And so my heart goes out to all of you. Also had the good fortune of being pretty well funded while I'm here. I listed some of my federal, philanthropic, non-governmental, and even internal JHU funding. I got to say, these pilot grants, from Lipitz to Fisher to CLF to so many different sources, they make some of these bigger grants possible, and we got to appreciate the opportunities that they create. Last slide, I want to thank my wife. Meeting her was another transformative event, and it's a transformative experience that's gone on now for more than 25 years. It's kind of hard to believe, but through all my ups and downs, she's been with me, and it's meant a whole lot. I want to recognize my two children. You saw them when they were little before. Now they're all grown up. My older son just got into college, and my younger son is a burgeoning electronic music artist, among other things. And I'm so proud of them. And they really make this worth it. And I also want to thank the people who I've played music with, gone to see music with, collected records with. We need a little spice in our life, and that helps a lot. And then lastly, I want to thank my bike buddies, my other means of staying sane when I'm not doing the things that I showed you today. And I promise I'll be back on the bike more when the weather gets warmer. So anyway, with that, thank you so much for indulging me today.
I'll go over there. Keeve, that was fantastic. Thank you so much. We have time for a couple questions online or in the room, and I'll grab the other mic. So Keeve, you can come back to the podium.
Okay.
Questions or comments? Over there, and then Charles. Well, for the people on Zoom.
Oh, right, gotcha. That was amazing. Thank you. I do have a question about when you reach some kind of pinnacle, like passing a bill or something. When you look at the world today, the environment we're in, how do you feel about keeping that information in the public's purview so that they can't reverse those decisions later on? How do you everyone, the public can't always be on top of the science all the time. So I'm always concerned with there's a disconnect between keeping the public informed of the issue enough that they can advocate for themselves, and yet not having to, so the public doesn't have to always be you.
Yeah. So Peter, thank you for coming, by the way. It's a great question. I think it's important to keep in mind that the public are one piece of the puzzle, and I don't like to rely on the public as the sole driver of change. I think when we build a coalition to pass a bill or foment some type of change, that coalition, I think, is what needs to be sustained until the issue is solved. And things are very rarely irreversibly solved. So I think continually mobilizing the advocacy community around the issue and then garnering the public's attention when there's a window of opportunity where their influence can be a part of making change, I think that's when you engage them. I don't like to burden the public unless we really think there's going to be a return on capturing their attention. And now's a tough time to capture the public's attention. I mean, I can tell you over the last couple of years, if we released the same study 10 years ago, we would have had an amazing media response to it. And now you can imagine there are any number of things that are going to be dominant in the news cycle, and it makes it a little bit harder to work with the public. So I think we need to be creative and strategic about when the public are part of our advocacy.
Great talk. And I gotta know, what's your favorite chemical?
So many to choose from, but I still love arsenic.
And so the systems approach that you described as part of this, that brings in both the engineering of public health as we go forward in this, in solutions, some of inadvertent consequences, but the idea of having the life cycle analysis that goes on these things, not only for the chemical antibiotics, but also for animal production and the reduction. How do you see this going forward in a way that we can really start driving change?
I think the focus on cumulative impact is an integrative way to pull everything together. And I think it allows us to look beyond a single system. So looking beyond animal production, looking beyond petrochemical production at the total burden that the human population experiences. And I think that framework integrates beyond a single chemical or multiple chemicals. And ideally, when we're able to advance the science more, we'll be able to think about how all the other life stressors that do render people more vulnerable to disease from any individual exposure they experience will be part of our decision-making process that will allow us to better protect public health.
Hey, Keeve, is this on?
Doesn't.
Well.
Thank you.
Done well. Thanks for a great talk, and your contributions to the field have been fantastic. But what I liked best about your talk was the optimistic vision of the future, that this is a time of change. And I wonder if you could just say a little bit more about that, especially for the students, because these are tough times for science, and yet it's tough times that bring innovation, change, and positive progress. So I wonder if you could say a little bit more about that.
I think that's exactly it, Tom. We, as students, as faculty, experience a lot of adversity and challenge. I don't think anybody makes it through this system without facing some really tough times. And we are in some unusually tough times for society. Used to be the case that when I thought work was difficult, I would go home and turn on the news and see other things going well in other contexts. And now it's been a little bit more challenging to do that. But I think our mission remains incredibly important. And despite all the threats to the way we do things, I think we need to get serious about reinventing ourselves and kind of thinking about the phoenix. Our old model of doing things in academia, we're never going to get that back. And maybe that's a good thing. Maybe the innovation that it's going to require us to mobilize is going to allow us to come up with better ways to fund ourselves, better ways to train our students in order to prepare them for the challenges the modern world poses. So I'm excited about the opportunity to be creative and to inspire people to use that creativity to advance their own careers. So I think we're in a transformation phase. I think that's really the way we need to look at this. And I think we're going to come out the other side even stronger.
There might be questions in this one. Somebody's on to turn the Zoom. Thank you. I think we're good. Anything with Oscar? Thank you.
This will be our last question. Hi, it's always great to listen to your talks. It's very inspiring and funny. I have two questions. First one is, so basically you developed based on your arsenic story and how you move all the way to the regulators to accept that, is you kind of developed SOP. So can you apply that, or are you planning to do the same, let's say for the dyes in the food, or antibiotics, or at least dyes, for example, which are prohibited in Europe for years? Maybe we can try to do it in the US as well. And my second question, if you have any doubles in your record collection, think of me.
Second one, I absolutely will, Etta. That's an easy one. I'll have to let you take a look. The first one, I love that idea. So you were saying an SOP for change, essentially, right? What a cool idea. And so I do want to make a call out to, I think Shelley Hearn's probably online, but her Center for Public Health Advocacy, they're working on things like that. What is the cookbook? And I don't think there's a very linear, simplistic cookbook. I think it's challenging and it depends heavily on the issues. But I do think strong science, advocacy, and public attention are certainly part of the equation in almost every setting. But I do think science comes first. You got to get the evidence right. We don't want to advocate for something that has a shaky foundation. But I do think the other elements of the puzzle are very important, and we can advance the methodology or the science behind effective advocacy and effective practice. Yeah, I think there's room for that.
Great. Well, wise words to end our time today. Please join me in thanking Keeve for his outstanding lecture. And again, thanks to everybody for making time to be here today. Keeve, thank you for just demonstrating the power of public health. And for me as dean, I love doing these lectures. I mean, I get to brag about our great faculty. So thank you so much. I also want to just acknowledge that some of Keeve's colleagues have invited folks to head out, be out here for some social time outside of this door. So I want to invite you to do that when we wrap up. So thank you again for being here. Thanks to everybody for being online. Take care and have a good evening. Thank you all.